All right, so today we're going to begin with the next topic, that is transcription. So, in the previous class, we were talking about how the DNA replication takes place, wherein we have seen that the newly synthesized DNA is formed taking the older DNA as the template. And that is why we call the DNA replication as semi-conservative, isn't it? All right, so today we're going to see the whole entire process of transcription and how it takes place. Now, see, transcription is the process wherein from the DNA molecule, RNA has to get synthesized, right? In like when we studied about the central dogma of molecular biology, we saw how transcription is the process from which from the DNA, the RNA molecule, particularly the mRNA molecule is synthesized. Now, in the prokaryotes, remember that the process of transcription, it takes place in the cytoplasm. And for the process of transcription, there is like the presence of special set of enzymes that we call as the transcripting enzymes. Now, the transcripting enzyme, it is present in the prokaryote. We call it as DNA-dependent RNA polymerase. Why DNA-dependent? Because it will take the DNA molecule and upon that it will make or synthesize the RNA. So that is why we call this enzyme as DNA dependent RNA polymerase. Understood? Now, during the process of transcription, there are 3 steps. What are the 3 steps? Initiation, second step is elongation, and then there is termination. Now, initiation is the step. Firstly, if you see over here, where is the picture? See, I'll show you the picture right now. See, during the initiation, first there is the initiation factor, which is called as the sigma factor. Okay, so this sigma factor comes, okay, and it binds at the promoter area, and then it will begin the transcription to take place. What will happen then? Slowly, the DNA-dependent RNA polymerase, it will start adding the nucleotides so that the RNA could be synthesized from the DNA. And it will move throughout the length of the DNA. And then the terminator or the termination factor would come, which we call as the rho factor, which will end this process of transcription. So this is like the pictorial representation. Coming on to the text, let us see. So first there is initiation. So RNA polymerase enzyme, it will bind to the promoter area and it will begin or initiate the process of transcription. Elongation, the second step, the RNA polymerase, it is going to use the nucleoside triphosphate as substrate and it will start polymerizing in the template dependent fashion. Template dependent fashion means on the DNA molecule. For example, if this is the DNA molecule and it has G, T, C, T, G, all the sequences, against this, the mRNA will be synthesized. Okay. And always remember the rule of complementarity will be followed. But instead of thymine, which nitrogenous base is going to be present in the mRNA, students?
Uracil.
Uracil, right? Everyone remembers this? So, during the process of transcription, the DNA helix is going to get open and then the whole entire DNA is going to get transcribed very nicely. Now, the last step will be termination, when this whole process will be coming to a halt or coming to an end. So once the enzyme, it reaches the terminator, then what happens? The RNA that is like formed later on, that will fall off along with the DNA, RNA polymerase. And that is how the process of transcription stops. Always remember there are 2 factors. RNA polymerase has 2 factors, sigma factor and rho factor. Sigma factor is the initiation factor that will cause the initiation, meaning it will begin the process of transcription. And the second is the rho factor, that is called as the termination factor. And what is it going to do? It is going to end the process of transcription. Is it clear to everyone? Now, always remember this one thing, students, that in the DNA, the adenine pairs up with thymine by 2 hydrogen bond. The guanine pairs up with cytosine with 3 hydrogen bond. This is for DNA. But in the RNA, adenine is going to pair up with uracil because RNA does not have thymine. And G will pair up with C. Okay, this must be on the tip of your tongues, and you should never forget this. Clear, students? Any doubts? Now see, this is the sequence that you've got. Okay, and from this sequence, you people have to tell me the sequence of the complementary DNA and the complementary RNA. Come on, write it and tell me. This is the parent sequence. And from here, you have to tell me the sequence of the DNA as well as the RNA. Jyobita, are you understanding this whole entire thing? Good. Find out the sequence and tell me what would be the answer. Done, students? Did you find out the sequence? Tell me what will be the sequence of DNA, the complementary DNA. Also, everyone switch on your video. Don't switch off your video. You will switch off when you will note down. Right now, you switch on. So what will be the sequence of the DNA? Someone tell me. See, adenine is there. Okay, you're not going to tell me, you're going to write in the chat box. Okay, I'll write it here. With adenine, you'll have thymine. With thymine, you're going to have adenine, then cytosine, guanine, thymine, guanine, then cytosine, adenine, thymine, and cytosine. Right? This is what everyone has got. This will be the sequence of the DNA from this template sequence. Now to find out the RNA, it will be the exact same thing. Instead, you're going to just substitute the T with the U. Isn't it, students? So you're going to be, it's going to be this thing. Right? This is how it's going to be. Is it clear to everyone? Did you people get how this is made? So always remember, in the RNA, there will be no thymine, there will only be uracil. So this was how the process of transcription is taking place in the case of the prokaryotic organisms. Now the RNA that is formed at the end of the transcription in prokaryotes, definitely it's going to be the mRNA. And remember one very important point, that it requires no modification. So at the end of transcription in the prokaryotes, there will be no modification that will be required. Now there is this question right in front of you, students. In case of bacteria, the process of transcription and translation, it takes place in the cytoplasm only. Why? Why not two separate places? Can anyone tell me? This requires basic knowledge. That's it. Tell me, why would it just take place in the cytoplasm? Why not any other compartment?
Is it because they're prokaryotes? They
So,
don't have all the stuffs.
Stuffs.
Like
They don't have stuff.
Proper.
You are at the right spot. Just answer it correctly. The way you're thinking is correct. See, since they are prokaryotic organisms, they do not have any compartmentalization in their cell, right? There is no boundary that is defining, okay, that this is the cytoplasm, this is the nucleus, this is that. They don't have that. So everything that will happen in them, it will happen in the main area, that is the cytoplasm. Understood? Is it clear to everyone?
Yes.
Okay. And remember that many a times, like for example, if this is the DNA molecule and from it the RNA is being it's just coming out only, then only the process of translation will begin in the prokaryote. Okay, it will like not even wait for the RNA or the mRNA to be fully transcribed. It will just begin then and there as it is. But when you compare the transcription of a eukaryote to the prokaryote, it's going to be very different. So remember, the process of transcription, like the basic steps are going to be the same. Initiation, elongation, termination, these things are going to be same. But in the case of eukaryotes, the process of transcription, instead of taking place in the cytoplasm, is going to take place in the nucleus. But it will be very complex. There are RNA polymerases that are present in the nucleus as well as inside the separate cell organelles as well. How many RNA polymerases are present? There are 3 of them. Okay, RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Now, RNA polymerase 1 of the eukaryotes, it will be transcribing, meaning it will do the transcription and form the 28S RNA, 18S RNA, and 5.8S RNA. Meaning these are the different types of RNA that are going to be synthesized by the RNA polymerase 1. And only the RNA polymerase 1 can synthesize them. After that, the RNA polymerase 3, it will be synthesizing the tRNA, then 5S rRNA, and SN RNA. That stands for small nuclear RNA. Lastly, there comes the RNA polymerase 2. Now, RNA polymerase 2, it will be doing the transcription of hnRNA, meaning this hnRNA further is going to make the mRNA. So, always remember the transcription that we study, that will be forming the mRNA at the end. It will be done by RNA polymerase 2. This is the name of the enzyme. Clear? And remember that this hnRNA, that stands for heterogeneous nuclear RNA, it is the precursor of mRNA, meaning before mRNA is formed, firstly the hnRNA will be made and then mRNA will be formed. Is this much clear to everyone? Does anyone have anything to ask? You can ask.
No, ma'am, no doubts.
See, once you people will read the notes and then when you read the NCERT, it will be very clear. Now, After the transcription is done, like once the whole entire initiation, elongation, termination, these steps happen, then what happens is that the mRNA that is formed, it will undergo a lot of modification. This was not taking place in prokaryotes, but the modification will take place in the eukaryotes. So after the transcription, the hnRNA formed, it contains exons and introns. Now remember, these are sequences, okay, coding sequences, useless sequences. So both the kinds of sequences are present. The presence of intron, it makes the RNA non-functional. So we have to remove the intron and then the exons only should be there. So there is this mechanism, like after the transcription is done, the first process that happens is called as the splicing, in which the introns are removed. All the non-functioning parts are removed from the RNA and the exons are joined. Apart from that, tailing and capping also happen. In capping, methyl guanosine triphosphate is added at the 5' end, meaning at the 5th prime end. The capping is done by methyl guanosine triphosphate. And then tailing is like at the 3' end, like at one end, methyl guanosine triphosphate is added, and at the other end, which is the 3' end, 200 to 300 adenylate residues are added. And now, after all of these modifications are done, now the hnRNA is fully processed, is fully modified, and now it is called as the mRNA. Now, mRNA, then it comes out of the nucleus and it comes out into the cytoplasm for translation process, or the other name for translation is protein synthesis. Is it clear to everyone? Any doubts? Oh. Now, the presence of introns is reminiscent of antiquity when it tells us that how this whole entire evolution has taken place. Also, the process of splicing represents the dominance of RNA world. This line NCRT is like direct NCRT line that tells us that RNA decides to remove the intron and keep the exons so as to make it functional. But if it doesn't want, it can become non-functional as well. Is it clear, students? So this was all there is to know as far as the transcription is concerned. After that, we have the various features of the genetic code. Okay, now genetic code, like how you people have seen A, G, T, all of that sequences that we make, 3 of them are together called as the genetic code. 3 of them, all 3 of them together. So that is what we call as a genetic code. So first of all, the genetic code is always in the form of triplet. 3 nitrogenous bases means one set or one genetic code. The second feature is that they have a comma-less nature, meaning you will read it without any punctuation. Like AUG will be read together. There will be no stoppage between A and U and G and like that. Third point is that the genetic code is degenerate, meaning there are some amino acids that are coded by more than one codon. For example, serine, leucine, arginine, methionine, these are some amino acids that get coded by more than one codon. So it's like they have multiple codons coding for the same amino acid. Then comes universal. Like remember that these genetic codes, they are universal for all the organisms. Meaning, if U, it is going to code for the phenylalanine amino acid, then right from bacteria up until the human beings, everyone will have U coding as phenylalanine. Is it clear? Then comes initiator codon or start codon. Always remember the AUG. It codes for the amino acid called as methionine, and it is also an initiator codon. So whenever there will be any process in terms of molecular biology that will have to start, it will start with the AUG. Clear, students? And
Yes.
lastly, the last feature of the genetic code is the stop signals or the terminator codon. Three of them, like UAA, UAG, and UGA, ochre, amber, and opal, they are the stop codons or the terminator codon, meaning that they do not code for any amino acid, and we call them as the nonsense codon, and wherever they will be present, they will stop the process. For example, AUG is there over here. It will start the process of reading. Then it will, like UUU will code for phenylalanine, then again another amino acid. But as soon as the stop codon comes along, the reading will stop then and there. It will end. So any of the stop codon, wherever it will be present in the genetic code, it will end the process. Is it clear to everyone?
Yes, ma'am.
All right. Okay. Now you people note down till here. You have to start from transcription, right? Hmm?
Ma'am, the proof of semi-conservative nature of DNA replication.
This part you've done, right? Types of mRNA, types of RNA also you've done.
Yes, types of RNA.
And DNA replication.
Yeah, we were doing the proof.
No, ma'am. We have to do this.
The proof. So I have to give you another writer. You people start writing from transcription, okay? This part.
No, we didn't finish writing the proof for semi-conservative nature of DNA replication.
This part you've not written.
The writings on the right side.
This part is this thing, DNA replication.
Yes, ma'am. We didn't do that.
We didn't do that.
You didn't do that. So DNA replication you have to do. Okay, you can note it down. Done, students? Done till here? Shall I scroll?
Yes, ma'am.
Everyone's done?
Yes, ma'am.
Done, students? Does anyone have any doubts up until now?
No, ma'am.
Is everyone done till here? Shall I scroll?
Yes.
1 second, ma'am.
Okay.
Done, ma'am.
Done, everyone? Shall I scroll? Students, are you done? Shall I scroll down?
Yes, ma'am.
Okay. Done, students? Shall I scroll down?
Yes, ma'am.
Done, students? Also, did you all understood the whole entire concept about transcription?
Yes, ma'am.
Is there any doubt in any of the step, any of the factor or the processing or why it takes place? Where it, like, do you people have any doubts regarding this?
Ma'am, I have a small confusion in the termination process.
Huh. So termination of prokaryotes, like in general, see what happens is that while the termination has to take place, meaning at that point of time, the RNA polymerase would have gone through the entire length of the DNA for making the RNA. Now the DNA is over. Now the RNA has to come out. So for the whole entire process of the transcription to end, there comes a row factor, which is the termination factor. And with the help of termination factor, the process of termination happens and the transcription then stops. And lastly, the mRNA is taken out.
Ma'am, the
Clear?
termination is like the last process where it concludes.
Which part?
The
Which part?
termination process.
Prokaryote or eukaryote? Last part as in like the RNA does not require any modification.
The enzymes involved on top. About this also.
Where here, transcripting enzymes.
The DNA replication part.
You're saying, you said specify termination of what? Termination
Okay.
of transcription or termination of replication? I was explaining transcription to you. See, the process of termination in the case of the DNA replication process, right? There is like once the whole entire parental DNA strand gets copied to like a new DNA strand is synthesized at the end of that, then what will happen? The newly synthesized DNA molecule is going to have the old parental strand and the newly synthesized strand. Now, when we talk about the enzymes that are found in the process of DNA replication, so once the termination is completed, the leading strand will be taken out as it is, but the lagging strand, the discontinuous strand, with the help of DNA ligase, they are going to be joined and made one DNA, like one long strand, right? So until now, in the whole entire termination process, the DNA helix that was unwound, now it will go back to its normal state and then it will basically like have a newly synthesized DNA molecule. Clear?
Yes, ma'am. Ma'am, we did this.
Done, students? Done till here?
Yes, ma'am.
Everyone's done? What about the others? Any doubts up until now? Anything you people want to ask me?
Are we supposed to like learn the diagrams?
Yeah, I will scroll towards the diagram also. First, you write the text part, then you go for the diagram. Okay. Done, students? Done till here?
Yes, ma'am.
Yes.
See how in this particular diagram you can see variety of the things. Like for example, first the the orange circular or sort of oval thing that you see, that is the RNA polymerase. And when that RNA polymerase combines with the sigma factor, which is the initiation factor, it goes there and binds with the promoter region of the DNA, and then the process of transcription begins. Now, slowly, slowly, it will start moving towards this side, right? And thereby transcribing the mRNA. Slowly, slowly, the sequences will be made, like how like sequences will be matching and the RNA will be coming out. Then this sigma factor will actually detach from the RNA polymerase, and then the rho factor, which is the termination factor, that will attach to the RNA polymerase and thereby end the process of the transcription. And this is what we call as termination. And at the end, the mRNA actually is coming out of the DNA. So utilizing the DNA template, the mRNA is being synthesized. That is why we say the enzyme used is DNA dependent, because the DNA is used as a template. So DNA dependent RNA polymerase. Is this diagram clear to everyone, students?
Yes, ma'am.
Okay.
Yes, ma'am. Ma'am, suppose this question comes like explain these 3 steps. Should we explain it with this diagram?
Yeah, if it comes for 5 marks, 100% you can draw the diagram to suffice your answer. And even if it comes for shorter and you feel like your answer is not enough, then you can draw the diagram always. In any kind of question, you can draw your diagram to support your answer. Okay, students, it doesn't have to be very perfect and colorful, but you can somewhat give the idea to the teacher, and always remember to give the captions. Right? Caption is very important. So here, for example, if you were to give the caption over here, you will write figure process of transfer, like that. Okay, always give the caption wherever possible.
Okay, ma'am.
Done, students?
No, ma'am.
Okay, okay.